changed. And if we make this recombinant DNA to produce multiple copies of the genes over and over again, which called gene amplification or cloning, this also process is now very well known and utilized in research in biomedical science. So many copies of genes, it's one of the very widely used application of this gene cloning amplification in polymerase chain reaction, real-time polymerase chain reaction and genetic analysis polymerase chain reaction. It's called gene amplification. This is a replication fork, origin of the replication in the bacterial cell. Very highly accurate. This is also origin of replication, and replication happened along the entire DNA of the entire chromosome of bacteria or DNA of the plasmid. Ribonucleic acid single-stranded, and the only differences between RNA and DNA that instead of thymine we have uracil as a nucleotide base pair of RNA. The same complementary base pairing to DNA molecules, except that of course uracil bind to adenine. The other versions of DNA, so messenger RNA is linear, one-stranded molecule. There is unique clover leaf-shaped transfer RNA and ribonucleic RNA that have spherical shape, forming major units of the, mainly forming the units of the ribosomes. Translation in prokaryotic organism happened just like it's happened in eukaryotic. The first of all formation using one strand of DNA as a template. Messenger RNA is formed based on the complementary base pairing, which with the help of DNA polymerase, starting with promoter, building on the DNA sequences. The movement again of messenger RNA is 5 to 3 direction. It's always the movement of the building strand. 5 to 3 direction. Transcription stops when the sequence, the growing messenger RNA strand reaching stop codon or terminator sequences. So this is first initiation, then expansion, and then with the help of DNA polymerase, and then termination. So we have the initiation of transcription, then a chain of messenger RNA is growing, and then we have termination. Codons, we already said that codons are the groups of three triplets, codons, nucleotides and messenger RNA that were complementary read from the DNA based on nucleotide base pairing and encoded for one amino acid. We have 61 sense codons, meaning that they in fact encoded for amino acid. Non-sense codons, two types, either stop codons that stop translation, or they are initiating codon, mostly accounted for methionine. 61 sense codons encoded for 20 amino acids. What does it mean? It means that there is a lot of redundancy, and for a reason. Because I started talking about mutation, pinpoint mutation, one amino acid was enough to change the protein synthesis, which will result in dysfunctional protein. Example to give you, sickle cell anemia. One amino acid and the whole red blood cells has prong shape. So the sense codon encoded for amino acid. Non-sense codon, methionine, start codon, adenine, uracil, guanine, and there is a few stop codons, again redundant stop codons, so that their translation could be stopped, because continuous translation beyond the stop point again result in dysfunctional proteins. Translation starts with assembly of ribosome. Ribosome have A, P, and E site for messenger RNA. Once messenger RNA is transcribed, it comes to the unassembled on the small unit or subunit of ribosome. This signals to large subunit to come and join in. They form functional ribosome with messenger RNA attached. The A attachment portion of the ribosome, then messenger RNA moves to P. You can A attachment, P protein or polypeptide. Usually first methionine comes to, which is start codon. Transfer RNA with methionine comes, and it has anticodon. So if we have here AUG, transfer RNA will have UAC, right? And based on base complementary principle, methionine gets attached. At the same time, the second transfer RNA comes here, reads the information based on anticodon, bringing another amino acid, and if this anticodon corresponds to codon of messenger RNA, the other amino acid gets moved to the P site where the polypeptides chain is building. The first transfer amino acid moves to E site, exit site, and moves out. The same happened with second and third, and on until polypeptide chain is building, unless it's come to the stop codon. Once to the P site comes stop codon, the last amino acid, last transfer RNA is removed from the ribosome, and polypeptide chain is ready. In eukaryotic cells, it mostly happened in the endoplasmic reticulum, the long polypeptide chains. In prokaryotic cells, it's happened right in the cytoplasm. Ribosomes is there, and they build the proteins right there. And very often the translation for prokaryotic cells, what is typical, for eukaryotic it's not possible while translation is not completed and proofreading of RNA not done completely. The building of proteins or transcription is not completed, and proofreading of messenger RNA is not done. The translation of building the protein is not possible. For bacteria it is sometimes. Translation still going, or transcription still going, and translation already started. It's another difference. Now exons and introns is very interesting concept. In small nuclear ribonucleoproteins or SNAPs, they can splice and remove exons from introns. So exons are regions of DNA that code for proteins, and introns intervening regions of DNA that don't code for proteins. Why do we need this? For proofreading, for make sure that we read correct information. It's one of the point of control. So DNA, exons, introns, the same, everything encoded here. But it's also weak point, because if there is no correct proofreading and intron stays, it's of course can be translated to the amino acid, and that again protein would be wrong. So those SNAPs RNA, they remove introns from the molecule of mRNA before it exits the nucleus and goes into cytoplasm or eukaryotic cells. forming only exons, leaving only exons as a sensible information material in the molecule. Again, the whole thing regulated by the genes, by the operons. There is two levels of gene expression control in eukaryotic cells. The pre-transcriptional control, transcriptional before messenger RNA is built, based on inducer bind to, by default, mostly the gene is constantly on, which is a repressible operon, right? So inducer and bind to transcription factor turns the gene on, and production is happened once the once enough protein post-translational control. When protein is enough, it's side of the operon. So the pre-transcriptional control, depending on what happening here, in the case of repressible operons, the product would be inducing the will be co-repressal, or it could be inducer in case of lactoperon. So, but in any event, it happened before transcription happened, pre-transcriptional. Post-translational depends on how much final product is there. Is it still need for it or not? And if it's not, then the final product based on feedback inhibition and then repressible operon function will shut the whole system off. In operon, again, promoter, operator, and operon, we already talk about this. This we also reviewed. It's the same from your textbook, which I already reviewed with you. So this you should be familiar with. The positive regulation is a catabolite repression, which is inhibit cells from using carbon sources other than glucose. It's catabolite repression. Cyclic amino acid build up when cells is not when cell is deprived of glucose, when glucose is not available. And cyclic adenosine monophosphate usually bind to the lactoperon promoter, unleashing this whole alactose or lactic operon induction, right? Inducible operon. I already mentioned this, that inducible operon or lactoperon happened with the, or activates with the help of cyclic adenosine monophosphate. This is glucose and lactose utilization by bacteria, and when there is no available glucose any longer, there is lag time that necessary to convert the normal glucose production by the catabolite repression to the lactic fermentation, which is lactoperon induction, and this is how it's all happened, and then it's converted to lactose use. This is again the same thing. And now I will stop here. We will review epigenetic control and histones during next week. And as always, review the material and send me email with your questions.